Hello everyone, this is Geethanjali. In today's video, we'll be learning about the Human Genome Project. So, Human Genome Project is a mega project which started in the year 1990 and ended in the year 2003. So, this project is associated with the development of a new area in biology called bioinformatics. So, bioinformatics is nothing but the study that focuses on collecting biological data and processing it. So, in this case, in Human Genome Project, so we are collecting the data of our genome and storing and processing it. So, what were the aims or goals of the Human Genome Project? So, first of all, it was to identify all the genes associated with the human genome and to find out the sequences of our 3 billion chemical base pairs, later storing this information and analyzing this data, and also to transfer this data to the sectors which could make some good use of it, and also to address the ethical, legal, and social issues that may come up while doing the project. So this Human Genome Project, as I told you, 1990 to 2003, so it was a 13 years old project, and it was coordinated by the US Department of Energy and the National Institute of Health, and its major funds came from countries like Japan, France, Germany, China, etc. And also the Wellcome Trust company from UK became the main partner. So apart from sequencing the human genome, there were some other non-human organisms like bacteria, yeast, Conorrhaptides, Drosophila, rice, etc., whose genome was sequenced entirely. So how exactly was this sequencing done? Sequencing basically means finding out the bases like A, T, G, C of the entire DNA. So how exactly was this achieved? So there were two methods. First is express sequence tag and sequence annotation. So there were major differences between these two methods. Sequencing the entire genome, including the coding sequences and the non-coding sequences, is basically referred to as sequence annotation. So you can see this is basically a blind approach as you are determining the coding and the non-coding sequences. So we don't usually have anything to do with the non-coding sequences. So just prefer not to sequence non-coding sequences too. But in the express sequence tags, here only the gene expressions which are transcriptionally active are sequenced. So that means only the coding sequences are determined. So how is this done? First of all, the DNA, whatever DNA it is transcribed. So we know only the transcriptionally active part codes for the mRNA, right? So the other parts are, that is the introns and all, they are removed during the post transcription modifications. So this mRNA which is there, this is again converted to DNA by the process of reverse transcription. So in this case now we don't have our introns etc. We only have the DNA that is transcriptionally active, or the exons. So for figuring out the basis, the DNA had to be cloned for easy sequencing. So in that cases, we need vectors, right? So the vectors used here were BAC and YAC, standing for bacteria yeast artificial chromosome, bacteria artificial chromosome or yeast artificial chromosome. So how exactly were these sequences determined is that we had these equipment, or rather I would say machines. These were called automated DNA sequencers. So these were able to determine the sequence of DNA. So there was a scientist named Frederick Sanger. Okay, he basically developed a method on the basis of which these automated DNA sequencers work. And he also had developed a method for sequencing the amino acids too. So he's credited for both sequencing the DNA and the amino acids. Moving on to some important features of the human genome. So after the project, after sequencing the entire human genome, they had the following data. So first of all, the human genome contained about 3,164.7 million base pairs. So you can see about 3 billion base pairs. And on an average, a gene would contain around 3,000 bases. And the largest gene in the human genome is the one coding for dystrophin. Okay, it's the dystrophin gene, which has about 2 million base pairs. Okay, and the estimated number of genes present in our genome is 30,000, and it is not as much as we had estimated. But anyways, the next point is that 99% of the nucleotide bases are exactly the same in all human beings. So you see 99% of all the human genome is exactly the same. Only that 0.1% which is there makes us different from each other. The functions of over 50% of the discovered genes are unknown. So, we have 30,000 genes, so more than 50,000 genes functions are not known. And in these genes, less than 2% actually code for proteins. Sixth point is that they have noticed a number of repeated sequences in human genome, and they are what that human genome makes up the most. So repeated sequences make up a very large portion of the human genome. These repeated sequences are repeated from 100 to 1,000 times, and they do not have any, coding functions, but they definitely help us in understanding the dynamics and structure and evolution of chromosome. And because of this, we came up with DNA fingerprinting. So we'll see that in the next video. Chromosome number 1 has the most genes, so about 3,000, whereas the sex chromosome Y has the fewest gene, only 231. And scientists have noticed in our genome, at about 1.4 million locations, there are differences in the base. Okay, and this difference is only one single base, and these are called single nucleotide polymorphism or SNPs. So these actually can be inherited, and that is why they are a great help in finding chromosomal locations for disease associated sequences, etc., and also tracing the history of a particular trait or disease in generations. So this was all the important stuff about Human Genome Project.